Life exists in great diversity. How is this diversity possible when all life is composed of the same basic components, like cells? The answer lies in genetics and genes.
Study of genetic makeup of a human and genes.
It's the passing down of, I don't know, nature versus nurture. I don't know how to put it into words. It's what my ancestors might have, the traits that I might take from them that might be similar.
The human body, the genetics of your genes, and your DNA.
It's like the link between like parents and children, and like I don't know, the study of like hereditary like genes and.
Well, genetics is a study of heredity, basically. That's it. How traits are passed on from generation to generation. That's genetics.
Well, a gene is what we have in our cells, our makeup, our body, sort of our whole structure from gene, from biological gene.
I did this in biology, but I can't remember. Just something that defines one of the characteristics, like habitually.
Something aware.
I don't have a clue.
Okay. So what is a gene? A gene is the basic unit of heredity. Basically, it's a part of a segment of DNA which codes for a trait which is heritable in an organism.
It seems we've got our work cut out for us. So let's start with the basics. What is a gene, really? In order to understand this question fully, first we have to look at things on a molecular level. DNA has 4 nitrogenous bases, with adenine bonding to thymine and cytosine to guanine. This pairing of the bases is essential to the construction of the DNA double helix. DNA is a double strand of nucleotides. A nucleotide is the repeating unit of the strand of DNA made of a phosphate and sugar backbone, with each nucleotide having a nitrogenous base. The sugar and phosphate are always the same, whereas the base varies between the already mentioned 4 different types. But now we run into a problem. The strand of nucleotides is too large to pass through the nuclear membrane to its destination in the cell cytoplasm. To get around this problem, we use a smaller molecule called mRNA to take down or transcribe the information from the DNA and transport it to the ribosomes, which synthesize proteins. Remember how the 4 bases like to form specific pairs with each other? This is exactly what happens between the mRNA and the single strand of DNA. The only difference here is that adenine bonds to uracil on mRNA instead of thymine. This is the process of transcription. This new strand moves to the ribosomes. Now every 3 bases on the strand corresponds to a certain amino acid, which are the building blocks of proteins. Then, through highly complex chemical processes, these chains of amino acids are converted into proteins. The enzyme lactase is a great example of how a malfunctioning gene produces a malfunctioning protein, which then has a negative effect on the organism. The enzyme lactase processes lactose, which is the main sugar present in milk. Now, if you have lactase present and it works, as in Oisin's case, drinking milk is grand. If, on the other hand, you've got a malfunctioning copy of that gene, like Connor does, drinking milk is something you might want to lay off, because you get wonderful things like cramps, nausea, vomiting, and diarrhea. So stand back, folks, we're going to do science.
So we're in a golden age of the era of sequencing. Has obviously heralded a golden age in genetics and microbiology, and the ease now with which bacteria can be sequenced, the variety of bacteria that can be sequenced, and the low cost of sequencing these bacteria has given us huge insights into the diversity of the bacterial world. We're in a golden age. There's a huge amount out there to be found out, and I encourage all of you undergraduates to head straight into molecular biology. Human diseases will be solved long before human bacterial disease will be we'll be able to even scratch the surface on that.